Good day, this is Professor Will's CBMD PhD. Today's Friday, May 29, 2020. It is 10:20:25 p.m. Eastern Standard Time, and this is an educational video based on biophysics. So let's get started here.
Introduction. The identification, purification, and separation of the components of a mixture of molecules is of considerable importance in molecular biology and biophysics. There are several techniques available for this purpose. The two most frequently used described in this chapter are chromatography and electrophoresis. Chromatographic techniques can be classified into four main categories based on the type of molecular interactions used for separation, viz. Ion exchange, chromatography, absorption chromatography, partition chromatography, and molecular exclusion chromatography. The techniques can also be classified in another way, on the basis of the mode of separation and the support system used, as column chromatography, thin layer chromatography, and paper chromatography. The particular technique that is used for a given sample depends on its characteristics. Sometimes several techniques may be used, one after the other, to obtain extra pure sample. The basic principle behind all the different types of chromatography is, however, the same. Chromatic separation takes advantage of the fact that the sample distributes or partitions itself to different extents in two different immiscible phases. This property is described by the partition or distribution coefficient K sub d. If we consider two immiscible phases A and B, K sub d equals concentration of the sample in phase A divided by concentration in phase B. The effective distribution is then defined as the total amount of the substance present in phase A divided by the total amount of substance present in phase B. The two immiscible phases could be a solid and a liquid, or a gas and a liquid, or a liquid and another liquid. Generally, one of the two phases is a stationary phase and does not move. The other is a mobile phase and moves with respect to the first. To understand how chromatography is used to separate and purify molecules, consider the column mode, with a stationary solid phase and a mobile liquid phase. To separate a mixture of molecules, a column is packed with the granular stationary phase to a height of a few centimeters and is then surrounded by the liquid phase. Assume that the column diameter is such that 1 centimeter cubed of liquid will surround 1 centimeter length of the column material. First, consider the behavior of a solvent containing a single solute. Let 32 Ug of the sample in 1 centimeter cubed solvent be added to the column. It would occupy position A. If the effective distribution coefficient is defined above as 1.0, the solute would distribute itself equally between the liquid phase, the solvent, and the solid phase, the support material, and the column. Now, if another 1 centimeter cubed of the solvent, this is also sometimes more properly called the eluent, especially when it is particularly used to elute or bring out the sample, is added to the column. Half the solute will move from position A to position B, since the distribution coefficient is 1.0. There will now be 16 Ug of sample each at positions A and B. If yet another 1 centimeter cubed of the solvent is added to the column, the situation will be as shown in the third panel, with 8 Ug in position A, 16 Ug in position B, and 8 Ug in position C. When this process is repeated, the pattern shown in the other panels will be observed. At every step of the equilibrium, 50% of the sample will be left bound to the solid phase at each position. After 5 equilibrium steps, the sample is present all through the column but has a maximum concentration at the center of the column. If the solvent contains two solutes with different partition coefficients at each step, the amount in the solid phase will be different for the two. After a number of such steps, the peaks of concentration for the two components will be at different positions along the column, thus achieving a separation of the mixture. If the process is allowed to continue sufficiently long, the different components of the mixture will flow out of the column at different times and can be collected separately. This is the basic principles of chromatography. We now consider different applications of this principle. Adsorption chromatography, partition chromatography, ion exchange, chromatography exclusion, chromatography, and affinity chromatography all use the column mode, where the stationary phase is packed into glass or metal column. The nature of the stationary phase depends upon a particular form of chromatography. It is generally an absorbent, in other words, a resin or a gel. In this case, a stationary phase is applied to a glass, plastic, or metal foil plate as a uniform thin layer, and the sample is applied to the top edge of the layer using a micropipette or syringe. This technique can be used for partition chromatography, absorption chromatography, and exclusion chromatography. The advantage of this method is that a large number of samples can be studied simultaneously. Also, it is quick and simple. In this technique, the cellulose fibers of a sheet of special chromatographic paper act as a support or stationary phase. There are generally two methods that are employed in paper chromatography: the ascending method and the descending method. In both of them, the solvent is placed at the bottom of the jar or tank, so that the chamber is saturated with solvent vapor. The chromatographic paper is held vertically inside the tank. In the ascending method, the lower end of the paper dips into the solvent and the sample spot is applied just above the surface of the solvent. As the solvent moves up the sheet of paper by capillary action, the sample constituents move along with it and get separated. In the descending method, the upper end of the paper where the sample is applied is held in a tuft of solvent at the top of the tank, and the rest of the paper is allowed to hang down without touching the solvent at the bottom of the tank. Again, by capillary action and gravity, the solvent moves down the sheet of paper, taking the sample along with it and separating the components. The components are detected by spraying the paper with specific coloring reagents. For example, ninhydrin, which binds only to amino acids and proteins. Sometimes fluorescent dyes are used. For example, lithium bromide is used with DNA sample. When the paper is subsequently examined under ultraviolet light, the position of the fluorescent or ultraviolet absorbing spots can be seen. The corresponding compounds are identified on the basis of their R sub F values, where R sub F equals distance moved by solute from the origin divided by distance moved by solvent. The R sub F value is a relative displacement of solute with respect to the solvent front and is constant for a given compound under standard conditions. Thus, by looking up the table of R sub F values, it is possible to identify the compound after separation. As mentioned earlier, chromatographic techniques are also classified on the basis of the molecular property of the support system or sample that is used in the separation. Some of these are considered in greater detail.
Good day, this is Professor Will, CBMD PhD, on infectious disease, biophysics, and microbiology. Well, hope you enjoyed listening. Until next time, have a great day.